The microbiome is the collection of all microbes that live in and on us. Research over the last several years from many groups has shown that the bacteria that live in our gut are interacting with the immune cells in our body. Furthermore, research has shown that the microbes in our gut somehow influence autoimmune diseases, which are chronic debilitating diseases where the immune system turns against itself and destroys the body. The exact mechanisms how the bacteria and which bacteria interact with the immune system to influence autoimmune diseases is largely unknown. We have studied a mouse model of an autoimmune disease called lupus. It is a systemic autoimmune disease, which means it affects many different organs. We found that when we changed the microbes in the gut of these animals with antibiotics, that we could prevent the disease and prevent the mice from dying from autoimmunity. We further found that specific molecules and cells that mediate this disease are prevented from damaging the body by treating the bacteria actually in the gut with an antibiotic. Most importantly, we found that specifically one bacterium did not stay in the gut but translocated, which means it traveled outside the gut into the liver and lymph node of the animal. And by doing so, it promoted autoimmune disease by stimulating the immune cells in the actual tissues. We further went on to show that actually a vaccine that we developed against this bacterium when given in the muscle prevented the disease from occurring by depleting this bacterium in the tissues and preventing it from traveling outside the gut into the tissues. Finally, we looked at human tissues from patients with the different autoimmune diseases that are reflected by this mouse model, most notably lupus, but also an autoimmune disease called autoimmune hepatitis, where inflammation in the liver occurs and the liver fails eventually. And indeed, we were able to find this bacterium in the liver of patients with lupus and autoimmune hepatitis, and also took human liver cells and cultured them together with the bacterium, which reflected the same changes and stimulatory effects on the immune system as we have seen in the animals. Finally, we saw that immune responses in the blood of the patients to these bacteria and certain particles of the bacteria were correlating highly with the autoantibodies that develop in this disease. Overall, we linked a gut bacterium from animals and humans with traveling through the body into tissues and thereby damaging the tissues and leading to autoimmune disease. This may have relevance for future new therapies that may target not just the immune cells, but the bacteria themselves, either inside the gut or even in tissues.